Morning, everyone. I'd like to welcome you to today's edition of the Royal Tyrrell Museum Speaker Series, sponsored by our Cooperating Society. So another shout out to them for sponsoring this and all sorts of other activities and initiatives here at the museum. So today we will not be talking about dinosaurs again. We're going to be talking about fish. So I'm wearing one of my fish shirts. And our speaker today is Jeff Liston. Some of you may remember Jeff. He was here for a few years during Femke Howalda's postdoc posting. And Jeff did some work here while he was accompanying Femke. So Jeff is originally from Scotland, as you'll probably tell from his accent later. After obtaining an undergrad degree in oceanography, evolutionary zoology, and invertebrate paleontology, you couldn't have just picked one, eh? At the University of Edinburgh, Jeff was involved in early work discovering the first dinosaur material and other fossil vertebrates on the Isle of Skye, off the northwest coast of Scotland in the early 1990s. Then Jeff did a PhD in vertebrate paleontology at the University of Glasgow on the Jurassic giant suspension-feeding fish Leedsichthys, a bony fish that was sort of the Mesozoic equivalent of today's filter-feeding sharks and whales. At the same time, he also worked on curating fossil plant and vertebrate collections from the Middle Jurassic at the university's Hunterian Museum. And during his time as a PhD student, he led a field excavation to collect the most complete skeleton of Leedsichthys, that giant fish, near Peterborough, the one in England, not the one in Ontario. After completing his PhD, Jeff moved to China, where at the Yunnan University's key laboratory of paleobiology, he worked on some of their early Jurassic dinosaurs. He then returned to Peterborough to curate the giant Leedsichthys skeleton he helped excavate 12 years earlier. Jeff is a founding member in 2003 of the European Association of Vertebrate Paleontologists, and he served as the president of that association in 2021. Last autumn, Jeff moved to the Netherlands with Femke at Utrecht University and is currently working on or exploring the delicate subject of fossil reparations and ethics and collecting and housing and all that fun stuff. Today, Jeff is going to be talking about descent into Carnivory, how large vertebrate suspension feeding in the Mesozoic ran out of options. So Jeff, you want to enlighten us about big fish?
Well, thanks for that, Jim. Okay, so this is the title, and I guess I should start off by apologizing. I'm kind of 12 years late because it was 12 years ago that I was first invited to come and do the speaker series here by Mike Newbury, who was then a postdoc of something called the Betsy Nichols Fellowship. Little did I know how much that was going to be a part of my life later on. And I visited in 2012, and so there's some of these remarkable things for those of you in Drum. This is Valley before Valley. I was just so fascinated by how much dinosaur material there was as part of the culture of the town, and I really liked it. In particular, this ballet dancing ornithopod from a place just up by where Tex Rex was, and everything was in the menu. Tex Rex done air. You can tell it was 12 years ago because gas is only $1.13. And so this was really quite a formative experience. So what we're going to talk about today is we're going to start by looking at suspension feeding, what it is. We'll look at some of the biological structures that are associated with this activity. We'll look for fossil evidence. We're going to have a little bit of a recreational field detour for some light relief. Look at some data that we get from the excavation, and then finally we're going to look at aspects of relatedness in terms of suspension feeders and their close kin. So, suspension feeding, the poster child, if you like, of suspension feeding is the whale. It's like this iconic animal, large, suspension-feeding, predominantly plankton-feeding animal. Currently, the blue whale, the biggest animal alive today, suspension feeder. So what is suspension feeding specifically? Well, fancy name is a form of biological hydrosol filtration. But the key thing is it's not selective consumption of food items. So, for example, the whale isn't aware of all the individual bits of plankton that it's eating. And hopefully the individual bits of plankton aren't aware of the whale, because that would be kind of a scary last few moments of your existence. And it involves basically these components of microphagy and very small prey and plankton consumption. Now, it's not the same as filter feeding, because that is a particular subset which involves a specific filter. And one of the problems that we have, of course, with fossil animals is it's very difficult to tell what the mechanisms are that they were using with their fossilized bones. So we generally just use the term suspension feeding. So we have extant living suspension feeders. And as I say, the water flows past some sort of feeding apparatus or structure. So, I mean, you look at some fishes, tadpoles, whales, even birds have these structures in their mouths. And it seems that it's quite a common strategy that groups of animals will adopt. Basically, if there's a good availability of planktonic food, then there'll be a tendency for a group of animals to adopt that as their primary foodstuff. So the sort of trends that we need to look for in the fossil record as well, projecting back from current animals, you have a high correlation of structures called gill rakers, rakers on the gill arches. And there's really quite a good trend of high gill raker counts and very elaborate or ornamented gill rakers as being indicators of suspension feeding. So in terms of things, there's things like digestive tract is lengthened, but obviously in a fossil animal, it's unlikely we're going to have that information preserved. So we would try and look for gill raker characteristics as indicators of suspension feeders. And corollary with that is usually there's edentulous jaws, which is an indicator that they're not using teeth to actually process food. So very quickly we'll look at gill rakers. They're ubiquitous amongst fish, but the specializations that we have for suspension feeders are very, very specific. So first of all, the generalized position. If you look at a fish, this is the primary gill lamellae. So these are the gas exchange structures, if you like, the equivalent to our lungs. Now, they're very soft tissues, very thin, very delicate structures, because the blood vessels need to be close to the surface for effective gas exchange. And that means that they're very vulnerable if the fish takes in water with a lot of grit or other particles that could damage these soft tissue structures. And so this is where gill rakers come in. They're kind of like baffles at the front. So if you see this here, these are the gill raker structures, and the idea is that they sort of catch any grit or problematic material and prevent it from damaging the gill lamellae. And if you have suspension feeding fish, these gill rakers become a lot more numerous, a lot longer, and a lot more ornamented, because they're doing more than just blocking grit and particles. So once again, to reiterate, there's this correlation of toothlessness. Generally, they're larger than their closer kin if they're suspension feeders, and also this increase in gill raker numbers and length. So let's look at some equivalents today. We have the basking shark, Ceterinus maximus, very common west coast of Scotland, and it looks a little bit terrifying, but of course the truth is that it's only scary if you're plankton. It's about a 9-meter length these animals go up to. And this is the sort of prey that they have, these copepods, usually a couple of millimeters long, very, very small animals. And the structure of the gill rakers, they may look like this massive gelled up hair, but if you take the individual particles out, it's these individual hair-like structures. This is the gill raker of Ceterenus maximus. But they come in all shapes and sizes. So this is the whale shark. And if you manage somehow, if these animals are washed up quite regularly on the Natal beach, if you manage to cut through the cartilage of these animals, then you find that they have a very different sort of structure here, if you close in on it. It's almost like a series of arrowheads. So they're all performing the same structure. They're a mechanism to extract plankton from the water that's coming into the mouth, but the mouths are different shapes, different velocities of water, so the structures are slightly different. Again, let's look at the eagle ray. This very elaborate structure, almost leaf-like when you look at it close up. These are the gill rakers. So let's see what we can get if we go back into the past and try and find something equivalent. And this really starts with a group of fish called the Pachycormids. Now, they're a Mesozoic group of fishes. They first really appear in the Early Jurassic. Generally, they're known from two main sort of horizons. The Toarcian Holzmaden shale, where you get these very famous for a lot of ichthyosaurs, and also the Solonophan Plattenkalk, where Archaeopteryx comes from. So that's Kimmeridgian, Tithonian, around Middle Jurassic Age. Generally, they're quite modest in size. They're usually less than a meter, and quite often they have these very long teeth, which we sometimes refer to as tusks, just because they project so far from the mouth. But not all of them are like that. This just shows you roughly where they are, just at the base, just before teleosts, which is the majority of today's bony fishes. This bony fish group is kind of different from teleosts. And I think in this regard, we have to talk about Leedsichthys. Leedsichthys is kind of like the exemplar of prehistoric suspension feeding animals. So this is the first figure that was ever published of Leedsichthys. It's 1890, a guy called Arthur Smith Woodward, who was kind of like the lead world authority on fossil fish at the time. And you think, oh, well, there's lots of bits of bone there, but actually, I have to stop you right there. Because the only specimens of Leedsichthys on this plate are these three components there. Everything else is from a different animal. And these three components, which I have to say were the only parts of this animal figured for the next 96 years, are gill rakers. And specifically, fragments of gill rakers. Now, why they chose those two, I don't know, because if you go through the rest of the type specimen, there's far more complete examples of them. But you can see that even though they're different style, they're very equivalent lengths to the rakers that Ceterenus maximus uses. Around 7.5 centimeters long is a good median. And you can see that they have a structure. So this base here is where they would attach to the gill arch in the mouth of the fish. And then you have these structures all the way along with a central groove. And we'll get a look at some of the details of that structure later. When was Leedsichthys is a good question, as Drax might say. Walking with Dinosaurs, I'm guessing you're all familiar with. The critical part in the development of this television series, Walking with Dinosaurs, took place when a pilot episode was done. And this was done through a collaboration between an animation studio who'd seen Jurassic Park and thought we can probably do that for cheaps as a sort of documentary style program. And Dave Martell, or the notorious DMM as he's sometimes called. And Dave worked with these animators and they decided to do a pilot for the series to see if it would work. And they chose a fauna called the Oxford Clay Sea fauna. It's a Middle Jurassic section, and it's all marine animals, including Leedsichthys and including these large ichthyosaurs called Ophthalmosaurus, Liopleurodon. And the reason why they chose this marine environment, because then you don't have to spend money on computer generating plants if you stick them all these animals are underwater. So this was the pilot episode based on the Oxford Clay fauna. And the Oxford clay fauna was predominantly excavated by this gentleman here with this wonderful suspension-feeding moustache, Alfred Nicholson Leeds, and as well as over 1,000 marine reptiles which he excavated from the clay himself. There was also a number of dinosaurs, like this, which was the most complete dinosaur for a while. And using a paleontologist for scale, this is the monument in the cemetery just by his local village outside Peterborough. And his brother, who also helped in the collection, had a similar monument in Auckland because he emigrated to New Zealand. So we know where the first specimen came from because there's a lot of archival material that's been kept by the family. So the old manor house here, Ibury, that's where they lived. And as boys, they'd go down to these little clay pits, including this one marked Tanholt. Today, it's overgrown, flooded, but in around 1885, they extracted the first bones. So it wasn't necessarily recognized for what it was at the time. This bone was found, and John Whittaker Hulk was looking at the collection of Alfred Leeds, and he said, "Oh, I know what that is. That's a stegosaur armor plate." And I know because I've looked at Marsh's papers on this. So at the time, in the 1880s, the theory was that stegosaur armor plates formed this continuous armor on the back. So that's what they thought this was. He was not correct, and the way that he found out he was not correct was because 2 years later, Othniel Charles Marsh actually visited Alfred Leeds' collection, and we can see this from the visitor's book, Yale College. And he said, yeah, I'm sorry guys, it's not a dinosaur, it's piscine, it's fish. So Arthur Smith Woodward was quickly sent off to work on this and describe it as a fish, which he hadn't recognized as before. But I have to say, this isn't the only time that Leedsichthys bones have been misidentified as stegosaur bones. These are the bones of the dorsal fin of Leedsichthys. And in 1901, von Huene was looking at some Leedsichthys bones in Cambridge, and he said, "Oh yeah, these are the tail spines of a stegosaurian dinosaur." Yeah, and that's what they were published as. But they were not. And one of the problems that we have here with this animal is it's very large, but it's very poorly ossified. A lot of the bones just never turn into bone. So, for example, they just remain cartilage. So, for example, there's no vertebrae have ever been found of this animal. Of the 70 individuals known, most of them are just a couple of individual bones, and that's it. There's only 3 or 4 specimens that are any more complete than that. And as a result, it's often been compared, particularly with the fragility of the bones, to looking at the anatomy of a conodont animal, which is very hard to work back from when all you're getting is these fragments. This was how I first came across Leedsichthys. This was a specimen in the Hunterian Museum, all 908 bones rolled out in the corridor. And the problem is that not a lot of those bones really resemble much of what a bony fish skeleton is supposed to look like. And a lot of that's due to the fragility of the bones. Once you excavate them today and remove the clay, they won't support their own weight and will just break while you're holding them. And this, as I've said, is due to the fact that a lot of the skeleton actually remains cartilage and doesn't ossify. And you can see this in other pachycormids. So, for example, this is a 1.7 meter fish from the Tuarcian Holsmaden shale. My grandmother was the last working fishwife in New Haven in Edinburgh, and she would probably have taken out the backbone very neatly all the way down there. But that didn't happen to this fish. That gap there is where the vertebra should be, and there ain't none. So we start to see this pattern even in animals that are less than 2 meters long, as pachycormids. This lack of ossification as a strategy, if you like. So another way of indicating these are the 2 bones that Whittaker Hulk thought were stegosaur armor plates. In 1887, when he published them, they were 80 centimeters long and 50 centimeters long. In 1889, when Arthur Smith Woodward looked at them, they were 64 centimeters long and 38 centimeters long. And by the time I got to them in 2005, they were only 42 centimeters long and 41 centimeters long. They just break and degrade even while they're just being stored over time. So you can imagine the perimeters and the shapes of these bones just gets lost. Another thing doesn't help. This is a note that you will find in the Natural History Museum in London for one of the specimens. And it refers to, there might be specimen parts of other specimens mixed in with this one. Brackets, accident in moving, January 1972. If you've ever been to the Natural History Museum in London and go to the stores, you'll notice that the lift on the fish floor is actually slightly offset from the floor itself. And the flatbed trolley that was carrying a whole load of those specimens tipped over. Causing Colin Patterson, the fish expert at the time, to say, I never liked that fish anyway. I've left out some of the more colorful parts of that sentence. So we have, if we look to the description of this, Arthur Smith Woodward, as I said, had been told to describe it because it was a fish, because Marsh said so, but he's clearly feels out of his depth. Within the description, there's so much hesitance. It's suggestive of, it's perhaps, it's probably, it seems like it could be. And this is really quite remarkable, and you can tell that he doesn't feel that confident about it. And you can go through it today and see what these bones are, but it was obviously quite a challenge for him. And I think we can reflect on this another way, which is the number of mixed misidentifications of Leedsichthys bones that have happened through time. So in 1882, a pectoral fin of Leedsichthys was misidentified as calcareous plant alga. And plant alga specialists were working on describing it until they realized it was a fish, and they were very disappointed. In 1886, as we said, some bones were misidentified as stegosaur armor. In 1889, some gill rakers were misidentified as fish ribs. Another identification as a sturgeon, that's a bit closer. A shark, getting cold again. Another publication said that they were tail spines, as I mentioned. And more skull bones were misidentified as a sauropod limb bone, if you can imagine that. Best one of all, in 1982, some fin rays were misidentified as being horsetail reeds, fossilized reeds, until they sent them to a paleobotanist, and he said, guys, there's Haversian canals in here. I think it's bone, I don't think it's a plant. And in 1998, some gill rakers were misidentified as being part of Pterodostro, which is a large suspension-feeding pterosaur from South America. And then again, a completely different bone was misidentified as stegosaur tail spines. So this indicates how problematic it is to identify this animal. And in fact, if you plot all of these, you'll actually see that Leedsichthys is not just a king among fishes, but perhaps a fish for all kingdoms in terms of where all these misidentifications lie. So what about specimens that actually look like what they are? And the tail specimen is probably the best example of that. This is a discovery in 1898, again by Alfred Leeds from the Oxford Clay. It's a complete tail of this animal. And this tail is 2.7 meters from tip to tip. So you start to get an idea of how big this animal was. Another indicator is the gill basket specimen. Again, Alfred Leeds excavated this, sold it to the Natural History Museum, July 1905. This gill basket is over 2.1 meters high, so that's the basket that lies in the mouth of the fish, and over 1.5 meters across. So this is a big animal that we're talking about. And within the gill basket, we see our beloved little gill rakers again. And if we zoom into these gaps between the arches, we'll actually see the rakers are overlying each other in echelon to provide this mesh which helps extract plankton. So in 2001, I was 1 year into my PhD, and that's the literature search year. And then I got sent a bone in the post, and some students had found this in one of the pits in Peterborough. And they sent me this fragment and said, oh, we think it's Leedsichthys, what do you think? And I could compare it with actually the bones that von Huene had said were stegosaur spines. It's like, yeah, this is a dorsal fin ray from Leedsichthys. So we went down there, and it was in the Star Pit just outside Peterborough. And this was actually one of the last pits that Alfred Leeds worked in before he died in about 1915. And we went down to visit the pit as a prospecting trip. That's Dave Martill again. And that gap there is about 8 meters. And we found about 12 different identifiable bones of Leedsichthys coming out along that stretch. And we started to get excited because, as I've said, a lot of these bones that we get for this animal, they're just like little isolated clumps. This might be a complete one. And we were foolish enough to think this would be a good thing. The problem was the bed had 22 meters of clay on top of it. So what do? Well, we came back almost a year later, and we had Komatsu, and we spent the next 3 days taking away the 22 meters of cliff until eventually we had this bed revealed here. And it's the redoubtable Dave Peppercorn who operated this machine. I remember at one time he just said, "Oh, do you want me to take another inch off with that?" And you could actually watch him and just scrape delicately an inch off to get it back down. It's amazing. Anyway, so you can see where the cliff used to be because there's a hole. And that was where we set up camp and started working. And this enters the period of 40 days and 40 nights in the pit where we excavated, or how I learned to stop worrying and live with the fish. So we lived on site. That's Mark Evans, some of you may know, plesiosaur worker, and Darren Naish as well, working there. And we had to work with dental tools to try and get these very delicate bones out of the clay. As you can see there, that's part of a plastic mapping sheet that we did to try and record the position of all the bones before we lifted them. And then at night, we'd have to cover it up with a tarpaulin so it didn't get damaged by the cold. And this was the extent of bone that we found on that bed from this one animal. So the bones stretched 23 meters across and then about 6 meters inwards, like that. So we had a lot of bone to excavate, and we didn't have much time because we were self-funded largely, and we did some night shift work as well. And some of the problems that we had was we had to camp at the top of the pit, and that was next to a railway line. And sure as anything, at 3 a.m. every morning, the freight trains would come through. And then, because the good old English weather, it would occasionally turn the pit into the Somme. And I don't know if you noticed the nice little bit of dip in the bed angle there, meant that all the water got caught. This is Peter Green, a retired surveyor, helping dig out the next morning. And we'd have to pump all the water out. And we would start with it like this, with all our little bags and everything of specimens, and then we'd have to pump it out, and then we could actually start digging again. Because of the delicacy of the bones, a lot of the bones were lifted in chunks with the clay. We developed a lot of little techniques with tissue paper and plaster and mixing them together that worked quite well to excavate large amounts of material. This is actually the entire left pectoral fin with a lot of other bones on top of it, so we couldn't really separate them out, so we had to lift it as one. Dave Martill thought this would be an excellent idea to use this polycell material. It's like froth pack. But as any of you will probably guess, this sort of material, although it goes off on the outside, it stays liquid on the inside. So all those delicate bones were inside, sloshing around and smashing on the inside. And that is why froth pack we refer to as the jacketing agent of Satan, because it really, really was a very, very bad material to be packing bones in. This is the left pectoral fin on the last day. We couldn't get it out any other way. We had to get the large excavator back in to lift it. And after about 3 months, we filled a 9-tonner truck with all of the bone material. We had about 2,100 bones that we'd extracted by this stage. The animal got called Ariston because, I don't know if you had the same ads over here, but it went on and on and on, and the bone just didn't stop. We had lots of new data, new bones, fixed physical juxtapositions, so this was really good because generally these finds had never been mapped. This is the skull roof of the animal, the bulk of the skull roof. These are the two opercles or gill covers, the left and the right, lying on their sides, so it looks very much like the animal came to rest on the seabed. And the left pectoral fin and the right pectoral fin, again, with this gap in the middle, again, looking like it came to rest on the seabed. So we did the usual things. We plastered pectoral fins, and we knocked them out and took the intact pieces away. This pectoral fin is 1.6 meters long. And Peter Green, our surveyor, retired surveyor, produced this sort of map as a sort of a simplified version of the 23 odd plastic sheets that we had covering the whole dig site to show us where the main bones were. So that's the left pectoral fin, right pectoral fin, some pelvics there as well, which is very unusual, and some of the skull bones going forward. So this came onto some publicity. And we had a little bit in the National Geographic. After a 1-hour phone call with Chris Sloan, unfortunately, that's the only quote that he decided to go with, but there we go. We had a reality show, which, they do those, we have to do such and such a thing, and we've only got 2 days to do it for made-up reason. They came in and did a show about, it was a paleontology series, and it was always, they had the 2-day Jeopardy thing, so they did one of those programs on the dig. And then we had the truly awful BBC reconstruction that was done as a result. I don't know if you remember, but it's like a chameleon, basically. I remember I was a consultant on this program, believe it or not. And I'll tell you what BBC consultancy works out as. We have... I got phoned up, and they said, okay, we've sent you the pictures, what do you think? And it's like, well, the pectoral fin's entirely wrong, you've made it like a whale, and you've done the same thing with the tail, the pelvics are wrong, you've made this thing look like a chameleon, it's awful. So there's silence on the other end of the phone, and they said, well, we've kind of built it now, so I think we'll go with what we've done. And then we get high-quality publications publicizing this as well. This is the Sun newspaper, double-decker bass. Again, going for the 35 meters long because they wanted to make it bigger than a blue whale. It's not bigger than a blue whale. And we also had a Journal of Creationism that cited the work and saying this is proof that the story of Jonah was real and was based on Leedsichthys. And one of my colleagues, Sarah Fielding, did this reenactment. But yeah, so that was 2005, and the specimen was donated to Peterborough Museum, and then we thought that would be it, until 10 years later. I was just finishing up my job in China when Peterborough Museum got in touch, and they said, we've got 66,000 to curate this specimen. We'd like you to come and be the project officer for this. Basically cleaning up the mess you made by giving us the specimen years ago. That's fair enough. So we went along and we started, and first thing I wanted to do was get all the mapping sheets laid out to try and make sense of where they all were, so we took it to the local Peterborough Theatre. The theatre wasn't big enough, so then we had to take the fish to the sports hall, and that was just about large enough to actually lay everything out mimicking the dig. And the reason why we needed to do this was we were going to photograph and digitize all of these sheets. And to their credit, I didn't think it was possible, but the website company did this. And if you go to the website, you'll find that they digitized the maps, the numbers are searchable with all the identifications of the bones, and you can zoom in, and you can also, apart from the skeleton that we have here, it's the first time anybody attempted that, you can grab individual bones and rotate them because they scan some of them, as I was saying to Ken earlier. And so it's a really, really good website. And we launched it in 2016, and very, very happy with the resources and everything. I urge you to go to it. And it was only a year or two ago when I was still here at the Tyrrell. I was talking to Ray Troll, who some of you may know, up in Alaska. He does a lot of fish reconstructions. He had actually done a painting of Leedsichthys years ago, and this is, for scale, he did the painting as Leedsichthys bigger than my house. So he's got it flying over his house, and that's him working at his board there. And I said to Ray, he says, you should go to our website, bigjurassicfish.com. And he came back and said, yeah, it's a gambling site. I said, what? And yes, a gambling site had squatted on our domain, and so this rather bizarre page is what you get. So it took some time, but then we managed to change it. So now, if you go to thebigjurassicfish.com, you get the actual website, and I commend that to you for all the resources and outreach material on that. It's really, really good. So getting back to the suspension feeding question, when we say a large suspension feeder, what are we talking about? Well, generally we're talking about an animal that's larger than a meter. So for example, we can look at this Carboniferous Permian animal that grew up to about 0.5 meter in length, and you can see these are all the nice long gill rakers that show that it was a suspension feeder, but technically it's still small. So when we say big, what we're actually meaning when we talk about a large fish. And I guess we managed with the material that we got from the dig to actually start doing some scaling. You might remember the black and white Cerostomous specimen that I showed you earlier without the vertebrae. That was a good animal to scale off. So we used different bones, some from Ariston, some from Glasgow specimen, tail specimen. And we managed to get a grid because we also did growth ring analysis so we could do the estimated ages of the animal. And the gill basket specimen came out as the largest at 45 years old and 16.5 meters long. Now, what does that mean? Well, looking quickly, there's very few data points, or there were at the time, for basking shark and whale shark growths. But the idea is that you say, well, maybe it's a similar trend that Leedsichthys is following. Who knows? But to show you how exceptional this is for bony fishes today, this today is really the longest bony fish that you can get. And you can see this is a king of herrings or an oarfish. And although it goes up to 11 meters, it's really not fish proportioned. It's more like a pencil with fins than anything else. And you really don't get a regular fish-shaped fish that's as large as 11 or 12 meters. So something different was happening back in the Mesozoic. And you can see that Leedsichthys is starting to be in the scale of either the very large chondrichthyans, like the whale shark, or heading into whale territory. So when I say 16 meters, how can we visualize that? Well, this 2-meter catfish is kind of similar to the black and white Cerostomous I showed you earlier. This is a 9-meter fish sculpture with people for scale. That's sort of basking shark size with humans next to it. But for 16 meters, this is a whale shark with somebody on a kayak for scale. So that's the size of bony fish that we're talking about. So imagine something a bit like that. So quickly do a bit about relatedness and cladistics with apologies. When I started my thesis, the most recent analysis of relationships within Pachycormids was one done in 1992. And it was only 15 or so characters and maybe a dozen taxa. So it was a very small data set. But if you look at Leedsichthys, you'll see there's only 1-2 characters actually scored for that animal. So my dipping my toe in the cladistic water, what I did for my thesis was I put some more characters in for Leedsichthys. I got this sort of pattern out which you can plot chronologically. And you can get this. This is a cluster of suspension feeders, so animals Leedsichthys, Cystinocormus, and Martolichthys, basically there. And then here you get the toothier, tusked, very carnivorous Pachycormids. And it's always a little bit odd that there's this big gap. This is an animal called Protosphorina, which we're about to come on to. But it seemed weird that there was this gap in the phylogeny. This is how you normally find Protosphorina. You get these, again, they're very large animals, but you don't get complete skeletons. You don't get vertebrae preserved. You just get disjointed fragmentary bits. Might sound familiar as another Pachycormid. And they have these interestingly scalloped leading edges to their pectoral fins. And for collection managers everywhere, Edward Drinker Cope once in one of his papers noted that for Protosphorina, This is a formidable weapon and could be readily used to split wood in the fossilized condition. I'm not exactly sure quite how he knew that you could split wood with a pectoral fin of a Protosphaerina, although I suspect that that pectoral fin is no longer with us. So these pectoral fins get very large. This is an American Museum of Natural History specimen called the Kaiser's Mustache. One thing that I found working in collections is that the larger specimens are the more easy ones to lose in collections because they don't fit in regular drawer systems. So they get stuck on top of cabinets or stuck to the side. If they're not on display, they're generally difficult to track down. So you can see here, this one is stacked across 4 filing cabinets. It was in a very compressed space, so I had to splice these 2 pictures together. That pectoral fin is 92 centimeters long. So basically, from that tip to that tip is taller than I am. So that's the size of Protosphaerina, the width, if you like, of animal that we're talking about. And to underscore the fragmentary nature, this is the only Protosphaerina specimen recorded that wasn't just isolated bits of bone. This specimen here, which you can see the head and the fins, and also these very long trailing pelvic fins all the way back. And that's really unique as a specimen. But from that, all the reconstructions that you see of Protosphaerina today with these long pelvic fins are basically derived from that one specimen. And basically, these pachycormids are behaving like billfish, like marlins or swordfishes. They have very long snouts. They're very fast and highly maneuverable. So this is another niche, if you like, the pachycormids sort of pioneered, was these billfishes, that form. Now, in 2010, just to round up the last part of relatedness, there was a little bit of a shake-up. The Niobrara chalk is where you get, I'll say, most Protosphaerina specimens. It's this sort of age in the Late Cretaceous, and there was 4 species of Protosphaerina present. And in 2010, a colleague of mine recognized that this one, Protosphaerina gladius, was actually a suspension feeder. So we jointly published this paper together and named it Bonerichthys gladius, and we sort of pulled it all the way out from Protosphaerina and thought, oh, isn't it amazing that people were confusing this with Protosphaerina for all these years. And so we changed then some of the ranges of suspension feeders. This, if you like, was my 2007 projection. And in 2010, we started adding in more suspension feeding pachycormids that were going basically all the way up to the end of the Cretaceous extinction. So that was suddenly showing that there was this lineage of suspension feeding that had gone on for a really, really long time. And again, we have these 2 main tribes, if you like, the tusked or the toothless ones that seem to be coming out on our phylogenies. And apologies for the resolution, but these very projecting sort of tusk-like teeth, if you like, for Protosphaerina. And you can see it in this cladistic analysis, which was done back in 2010. You get these main clusters. Here are the suspension feeders, and here are Protosphaerina and the main sort of tusked, toothy ones. But there was a problem with this because when I looked into the data set a few years later, I found that about 10% of the characters that were being used were all about tooth presence. And I got a little bit nervous about this because on the one hand, if you're a suspension feeding fish, the chances are you're not going to have any teeth, and therefore you're being, if you like, discriminated against in terms of the characters supporting your part of your clade as opposed to the other ones. And I wondered if there was a rescoring, reinforcing by sort of rescoring the same character multiple times. A few papers had come out since then which were talking about the unreliability of using teeth in cladistic analysis. And so I went back and took a look at this. And one of the things that prompted me to do this was during the dig, where we were digging Ariston out, we managed to get this really long maxilla out. And you'll see this was Big Meg. This is the Glasgow specimen. And it's a nice smooth surface there, 695 millimeters long. But this one is over 1 meter long. And here you have 5 or 6 teeth. So suddenly your suspension feeders, if they grow large enough, start to develop teeth. So that starts to break down again this division between the toothed, the tusked, if you like, Pachycormids and the suspension feeders. And then you might think that's a little bit odd. Why would a suspension feeder need to develop just a few teeth? But this tied into a discovery that had been reported in 1988 in Switzerland. This is a tilted seabed from around the Colovian time, so the same time as Leedsichthys. And these marks are interpreted as feeding traces, so animals coming down and feeding on the seabed and basically taking shellfish up. And there was a number of animals that were proposed for this. But one of the feeding tracks was different from the rest. It was about 1 meter across, and it was this sinusoidal pattern which you would get if it was a fish feeding along the seabed. And so a few people had speculated maybe this could be Leedsichthys. So in a way, finding these teeth on that jaw made perfect sense for an animal to be feeding like this, in the same way as walruses and grey whales do when they go down in the Bering Straits today. So I took a look again at this data set. I took out all the teeth, a little bit of dental extraction, and then I ran it again as an analysis, and Protosphorina leapt over to the suspension feeding group, and it's solidly nested within there. And there, to be fair, there's a lot of connections between the suspension feeders like Leedsichthys and Bonorichthys and Protosphorina in terms of their pelvic fins, their tails, the structure of the skull. There's a lot of things that link them together. So we have this interesting pattern where these disparate animals, and yeah, this is an old Leedsichthys reconstruction without the pelvic fins. This was a wedding cake topper of Leedsichthys, because obviously that's what you're going to do, right? And it actually shows the up-to-date long pelvic fins that we have for this animal. So it seems that Protosphorina is actually part of a secondarily carnivorous offshoot of the suspension feeders. The final thing, this was a thing I had in my mind, was if I was looking for something that linked Protosphorina to suspension feeders, what would I look for? And I thought, well, one of those nice pectoral fins on a suspension feeder, a little bit younger in the Cretaceous would be nice. And this specimen, which is going to be coming out later this year, it's a very early one before it was fully prepped out, is from the early Cretaceous in Australia, in the Eastern Interior Seaway. And it is a suspension feeder, and it has these wonderful little Protosphorina scallops all the way down the leading edge. So if you like, it's that link between these two groups. So conclusions, I'm sure you'll all be relieved to hear. Pachycormids pioneered the large vertebrate suspension feeding niche that is occupied today by chondrichthyans like the basking shark and the whale shark and baleen whales. And we can tell this because of their feeding structures in the fossil record. It's also an indication of the fact that they're way bigger than any bony fish today. Shows they had an efficiency within that environment which bony fish really haven't quite matched in the same way. And also it's a good indicator of the health of the oceans, because if you've got productivity that will sustain such large suspension feeders, you've got very, very healthy oceans. These macropredatory pachycormids like Protosphorina arose secondarily from suspension feeders and are secondarily carnivorous, which is quite unusual. And it's a combination of these characters supports the inclusion of Protosphorina within suspension feeding. And it's one of those things where you sometimes you hear stories about people who are researchers and they do their PhD and then they spend the next 15 years trying to correct all the mistakes they did in their PhD. And it's I have to think that back in 2010, when we took Bonerichthys out in isolation, if we'd actually spent a little bit more time thinking about, maybe we would have actually taken the whole group of Protosphorina out at the time and taking it in rather than doing it now. But what's done, it's done. And the last question remaining is, why is it that Bonerichthys goes to the end of the Cretaceous, but Protosphorina stops short by a stage, so the carnivorous one doesn't really make it? What else was a problem in that carnivorous niche in the sea? Well, I blame the mosasaurs, frankly, because they come in around then, and I suspect that they gave Protosphorina a little bit of a hard time, and no matter how maneuverable you were as a little billfish back then, I suspect you couldn't have competed with the mosasaurs, which of course leads to Femke's talk just last week. So with that, I would like to thank the Tyrrell Museum Cooperating Society, not just for helping us come here, but also for hosting us for 3.5 years. It was really a wonderful time, and it was we came away with a few extra things that we didn't turn up with, and including some wonderful guinea pigs, one of whom sadly we spread the ashes of last night. But finally, some stats: 3,100 hours of excavator time to dig the fish out, and we got 2,300 bones. And then after that, again, all voluntary, 11,750 hours of voluntary preparatory time of those bones at Peterborough Museum. And then all the digitization project for the Esme Fairbairn Foundation for the museum. And these co-authors, Anthony Maltese, Lavisa Retman, Mark Evans, Soledad Gurich Cavai, and of course Aperture Labs, who got me through my thesis. And with that, I say thank you, and if there are any questions, I'll be happy to give them.